Good afternoon, people.
Hi, ma'am.
Good
Hello.
afternoon, ma'am.
Good
Hi.
afternoon, ma'am.
Good
How are
afternoon,
you?
ma'am.
Good afternoon, ma'am.
Fine,
Good
ma'am.
afternoon, ma'am.
Good
Very good
Hello,
afternoon.
ma'am.
afternoon. Good afternoon. ಹೆಂಗಿದ್ದೀರಾ ಊಟ ತಿಂಡಿ ಆಯ್ತಾ
Hello, ma'am.
Hello. Yes.
Hi, ma'am.
Look
Good
where am I
afternoon,
standing
ma'am.
today. Can you guess where am I standing today?
Kutub Minar.
Where is Qutub Minar?
Kutub Minar.
Kutub Min-
Kutub Minar.
Where is it?
Ma'am,
Shifted to Bangalore?
Daily.
no, ma'am.
Delhi na? Allro Qutub Minar is in Agra, no? Who said Delhi? Shifted to Delhi also? There.
Delhi.
Why are you so underconfident about Delhi? Check. Go and find out where is Qutub
Delhi.
Minar. ಅಲ್ಲ ಬಯಾಲಜಿ ಅಂತೂ ಕರೆಕ್ಟಾಗಿ ಮಾಡಲ್ಲ ಅಟ್ಲೀಸ್ಟ್ ಜನರಲ್ ನಾಲೆಜ್ ಅರೇ ಪ್ಲೀಸ್ ಐ ಎಕ್ಸ್ಪೆಕ್ಟ್ ಯು ಟು ಅಟ್ಲೀಸ್ಟ್ ಗಿವ್ ಮಿ ಅ ಪ್ರಾಪರ್ ಜನರಲ್ ನಾಲೆಜ್ ಆಗ್ರಾ ಬ್ಯಾಂಗ್ಳೂರ್ ಆರ್ ಡೆಲ್ಲಿ ಎಲ್ಲಿದು ವೆರ್ ಇಸ್ ಇಟ್ ರೈಟ್ ನವ್ ಇನ್ ಬ್ಯಾಂಗ್ಳೂರ್ ಬಿಕಾಸ್ ಐ ಆಮ್
Delhi.
ಸ್ಟ್ಯಾಂಡಿಂಗ್ ಇನ್ ಫ್ರಂಟ್ ಆಫ್ ಇಟ್ ಡೆಲ್ಲಿ ಶೋರಾ
Delhi, ma'am. Delhi, ma'am.
Okay, okay,
Delhi.
okay. Okay, fine. What we did last question solve ಮಾಡಿದಿವಾ we did na translation ಮೇಲೆ question ಆಗಿದೆ correct so we have to start with regulation
ma'am.
of
Yes,
gene.
ma'am.
Yes, ma'am.
Regulation of gene. What is regulation? What is the meaning of term regulation? Regulation ಅಂದ್ರೆ ಏನು ಎನಿವನ್ ಈಗ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಐ ವಾಂಟ್ ಟು ಸ್ವಿಚ್ ಫ್ಯಾನ್ ಆನ್ ಆಗಿದೆ ಫ್ಯಾನ್ ಸ್ಪೀಡ್ ಕಡಿಮೆ ಮಾಡಬೇಕು ಅಥವಾ ಫ್ಯಾನ್ ಸ್ಪೀಡ್ ಹೆಚ್ಚಿಗೆ ಮಾಡಬೇಕು ಅಂತ ಹೇಳ್ತೀನಿ ವಿಚ್ ಮೀನ್ಸ್ ದ ಬಟನ್ ದಟ್ ಈಸ್ ಗಿವನ್ ಆನ್ ದ ಫ್ಯಾನ್ ಟು regulate the speed, that is called regulation. Regulation doesn't mean something to switch off or something to switch on, it is means to control a process. Regulation meaning to control a certain Ranebendur girls settle down. Why Ranebendur class 2 is talking so much? There is no one in Ranebendur class 2. Ranebendur class 2. Why? Okay. Fine.
I'm
So,
struck ಆಗುತ್ತೆ ma'am.
Person.
Screen is struck, ma'am.
screen is not stuck. I am not speaking anything. Hold on. Okay. Now it is moving.
Yes, ma'am.
Yes. Now it is clear.
ಇದು ಒಳ್ಳೆ ಐಡಿಯಾ ಅಲ್ಲ ಸಿಂಪ್ಲಿ ಸ್ಟ್ಯಾಂಡ್ ಲೈಕ್ ದಿಸ್ ಅಂಡ್ ಅಸ್ಯೂಮ್ ಇಟ್ಸ್ ಸ್ಟ್ರಕ್ ಗುಡ್ ಐಡಿಯಾ ಓಕೆ ಫೈನ್ ಸೊ ವಾಟ್ ಈಸ್ ಅ ಮೀನಿಂಗ್ ಆಫ್ ದಿಸ್ ನೌ definition what is the definition of operon
Such arrangement
present
is
in
very
E. coli
common
bacteria.
and regulated.
wait one of you which class is speaking is it Chikkodi
Chikkodi.
class 20
Yes,
class
ma'am.
20 Chikkodi is it you
Yes, ma'am.
huh Chikkodi class 20 you only read now definition of operon what it is given book ಅಲ್ಲಿ ಓದ್ ಹೇಳ್ರೋ it's there in book also what is given karma
Arrangement of
The system
bacteria.
of gene regulation comprising of a promoter gene, a operator gene, structure genes, and a regulated gene.
operon ಅಂದ್ರೆ ಏನು ಹೇಳು ಸಾಕು ಧಾರ್ ಹಾ
Arrangement of bacteria.
ಕೇಳಿಸ್ಲಿಲ್ಲ ಧಾರವಾಡ anyone from you what is operon
Gene present in E. coli bacteria.
ಅಷ್ಟೇನಾ ಓಕೆ ವಾಟ್ ಇಸ್ ದಿಸ್ ದೆನ್
The complete unit of genes comprising.
I have told you the meaning of polycystronic remember what is monocystonic and polycystronic
Yes, ma'am.
people see if you keep on forgetting the previous things no ಈ ಜನದಲ್ಲಿ ಮುಗಿಯಲ್ಲ ಈಗ ಚಾಪ್ಟರ್ ಹಂಗಂದ್ರೆ ನಾಳೆ ಎನಿ ಕಾಸ್ಟ್ ಐ ಆಮ್ ಗೋಯಿಂಗ್ ಟು ಫಿನಿಶ್ ಆಫ್. ನಾಳೆ ಯಾಕೆ ಎಚ್ ಜಿ ಪಿ ಆ್ಯಂಡ್ ಫಿಂಗರ್ ಪ್ರಿಂಟಿಂಗ್ ಎರಡೇ ಕಾನ್ಸೆಪ್ಟ್ ಉಳಿದಿದೆ ಆಫ್ಟರ್ ದ ಒಪೆರಾನ್, ಆ್ಯಂಡ್ ಇಟ್ ಈಸ್ ವೆರಿ ಈಸಿ. ಐ ಆಮ್ ನಾಟ್ ಗೋಯಿಂಗ್ ಟು ಡೀಟೇಲ್ ಆಫ್ ಎಚ್ ಜಿ ಪಿ ಆ್ಯಂಡ್ ಫಿಂಗರ್ ಪ್ರಿಂಟಿಂಗ್. ಇಟ್ ಈಸ್ ಈಸಿ, ನೀವೇ ಓದ್ಕೊಂಡು ಬರಬೇಕು. ಐ ಆಮ್ ಗೋಯಿಂಗ್ ಟು ಸಾಲ್ವ್ ದ ಕ್ವಶನ್ಸ್ ಡೈರೆಕ್ಟ್ಲಿ. ದಿಸ್ ಇಸ್ ಅ ಕಾನ್ಸೆಪ್ಟ್ ಬೇಸ್, ಅದಕ್ಕೆ ಕಲಿಸ್ತಾ ಇದ್ದೇನೆ ನಾನು. ವಾಟ್ ಈಸ್ ಪಾಲಿಸಿಸ್ಟ್ರಾನಿಕ್ ಆ್ಯಂಡ್ ಮೋನೋಸಿಸ್ಟ್ರಾನಿಕ್? ಕಮ್ ಆನ್, ಡು ನಾಟ್ ವೇಸ್ಟ್ ಟೈಮ್.
Single.
Synthesis of lac mRNA molecules continuously is called polycistronic.
ಹಂಗಲ್ಲ. ಓಕೆ, ಹೂ ಆನ್ಸರ್ಡ್ ಮಿ?
Gene.
ಐ ಜಸ್ಟ್ ಕರೆಕ್ಟ್ ಯು ಲಿಟಲ್ ಬಿಟ್. ಹೂ ಆನ್ಸರ್ಡ್ ಮಿ ದಿಸ್?
Ranebennur.
ಹಾಂ, ಈಗ ನೋಡು, ಇಫ್ ಯು ರೀಕಾಲ್ ಫ್ಯೂ ಕ್ಲಾಸಸ್ ಬ್ಯಾಕ್, ಮೋನೋಸಿಸ್ಟ್ರಾನಿಕ್ ಆ್ಯಂಡ್ ಪಾಲಿಸಿಸ್ಟ್ರಾನಿಕ್ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಮಾಡುವಾಗ ಹೇಳದೆ, ಮೋನೋಸಿಸ್ಟ್ರಾನಿಕ್ ಮೀನ್ಸ್ ವಾಟ್? ಅ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಯೂನಿಟ್ ವಿಚ್ ಹ್ಯಾಸ್ ಓನ್ಲಿ ಒನ್ ಜೀನ್. Polycistronic means what? A transcriptional unit which has many genes. Now, what is transcriptional unit? Now, everybody, okay, keep pin drop silence and all your eyes and ears on the board, because this will be my last time to make you understand this. Aridadne tirga same thing I am repeating now. Polycistronic means what? A unit having multiple genes or many genes. So, in case of many genes, transcription unit alli ಏನೇನು ಇರುತ್ತೆ? What does a transcription unit has? It has promoter, operator, genes and repressor. This whole thing consists of one transcriptional unit. When in one unit there is only one gene present, if this one gene gives only one protein, then such kind of unit is called as monocistronic. When in a gene there are in a transcription unit there are multiple genes, maybe 3 to 4, 1, 2, 3, 4, and you get 4 different polypeptides from this, 4 different proteins from a one transcriptional unit. This is called polycistronic. Did you recall this? Idan agide. I have done this earlier also. Do you recall this now? Is this
Yes,
concept clear? Monocistronic and polycistronic?
Yes, ma'am.
Yes, ma'am.
So now gene regulation at translation level we are studying for prokaryotes. The term operon means what? Cluster of genes. It is a cluster of genes that are having a one common operator and one common promoter, and that's why we say it's a polycistronic gene. ಈಗ ನಿಮ್ಮ ಬುಕ್ಕಲ್ಲಿ ದೇ ಹ್ಯಾವ್ ಗಿವನ್ ಲ್ಯಾಕ್ಟೋಸ್ ಆರ್ ಲ್ಯಾಕ್ ಓಪೆರಾನ್. Operon is what? A polycistronic gene that has a common promoter. What is it? Common promoter? ಏನೇನಿದೆ? Promoter, regulatory gene, operator, then what are the 3 structural genes?
Structural gene.
Which 3 structural genes? 3 genes. Which
ZY.
Like ZY.
Okay. Now, this whole thing is called as one transcriptional unit. And in one unit, you have 3 genes. That's why polycistronic. Andre, onesate polymerase, when it starts transcribing, it will transcribe all 3 genes. Bare Z madbittu nilatte anta hangagalla. No, polymerase will give all 3 genes and all 3 products. That is why it is called as polycistronic. If a gene has only promoter, operator, and only some X gene, let's say, ishte, ಒಂದೇ gene ಇದೆ, and ಈ ಒಂದು gene ಇಂದ ಏನಾದರೂ protein ಬರ್ತಾ ಇದೆ, this is called as this arrangement of such monocistronic. Correct. Okay, now this concept clear? Illivargu clear now? Okay. Now, this all is happening in whom? This is happening only in prokaryotes. Eukaryotes alli agalla. Why? Because eukaryotes have only one gene. They are monocistronic. Prokaryotes have multiple genes in a one unit. So, polycistronic. That is why the gene regulation of lactose happening in only, or the operon concept is found only in prokaryotes, never in eukaryotes. Okay. Now, Lactose operon is the only one example that is given in your book. Apart from that, there are multiple examples also. Tripoperon, arabinose operon, many different operons which are responsible for regulation of that particular substrate. Lac operon regulates lactose. Tripoperon regulates tryptophan. Arabinose operon will regulate arabinose. So different things, different products. Let us go with only one example now. P does what? What is P?
Promote.
Correct. Boys, none of you are speaking, okay? I want you to interact. All class boys, except Ranebenur. Ranebenur is a girls, okay? Others, Chikkodi and Nippani, you are Dharwad also. Boys are there in the classes, right? I want you to speak now. ಅಲ್ಲ ರಾಣೆಬೆನ್ನೂರು ಬಿಟ್ರೆ ಚಿಕ್ಕೋಡಿಯಲ್ಲಿ ಬಾಯ್ಸ್ ಇದ್ದಾರೆ.
ಮ್ಯಾಮ್ ಇನ್ ಧಾರವಾಡ ಆಲ್ಸೋ.
ಅದಿಬ್ರು ಜನ ಕೂತಿದಾರಲ್ಲೋ, ದೇ ಆರ್ ನಾಟ್ ಬಾಯೋ ಕ್ಯಾ?
ದೇ ಆರ್ CS ಮ್ಯಾಮ್.
ಓ ಬಾಯೋಗೆ ಬಾಯ್ಸೇ ಇಲ್ವಾ ಧಾರವಾಡಲ್ಲಿ? ಹಾ?
Only one is there.
ಯಾರು? Is he there today?
Yes ma'am.
Give him the mic. Who is that lucky person?
Lucky.
Yes, Are talking to me is lucky, you know. I'm standing in front of Qutub Minar today. Tell me your name.
Muhammad Arif. Ma'am.
Muhammad? Munde?
Arif.
Arif, okay. You are the alone biology student?
Yes, ma'am.
Others? CS, huh? Okay, do
Yes,
you
ma'am.
understand what we are doing? Ivatte matadadu. After 3-4 months, I'm talking to you today. Do you understand what we do in the class?
Yes, ma'am.
Okay, I want you to keep on speaking to me. There is only one mic in the Dharwad class? Okay, one mic, Arif, you only keep talking. Chikkodi class 21, boys, yell the mic. Where is the mic? I'm not going to teach, hinge nindu bidtini. Struck. New technique. I have got this. Helode illa, struck. Chikkodi boys. Class 20, class 21, eradu. Boys shift. Chikkodi class 20 boys ellaru in between bench shift agi. Everybody shift.
Hello.
Shashank sir made them shift. Shift quickly and take the mic. Don't waste my time.
Hello.
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 Who is that? Nonsense. ಈಗ ನಿಮ್ಮ ಹತ್ರ ಮೈಕ್ ಇದೆ. ಶಶಾಂಕ್ ಸರ್ ದೇ ಹ್ಯಾವ್ ಮೈಕ್. ಬಾಯ್ಸ್. ಗರ್ಲ್ಸ್ ಇಲ್ಲಾಂದ್ರೆ ಪಾಸ್ ದ ಮೈಕ್ ಟು ದೆಮ್. ಚಿಕ್ಕೋಡಿ ಕ್ಲಾಸ್ 21, ವೇರ್ ಇಸ್ ಯುವರ್ ಮೈಕ್?
Yes ma'am.
Zoom class 21.
Hello.
Zoom class 21.
Hello ma'am. Hello.
ಎಲ್ಲಿದಿ ನೀನು?
ಶಶಾಂಕ್ ಮಾತಾಡ್ತಾರೆ 20 ನಂಬರ್.
ಸರ್ ಪ್ಲೀಸ್ ಗಿವ್ ದ ಮೈಕ್ ಟು ಬಾಯ್ಸ್ ಆಲ್ಸೋ.
ಮ್ಯಾಮ್ ಇದು ಒಂದು ಮೈಕ್ ಪ್ರಾಬ್ಲಮ್ ಆಯ್ತು. ಒಂದು ಸ್ವಲ್ಪ ಸ್ಲೋ ಮಾತಾಡಿದ್ರು ಇಲ್ಲಿ ಕೇಳಿಸಂಗಿಲ್ಲ ನಿಮಗೆ. ಪೂರ್ತಿ ಜೋರಾಗಿ ಮಾತಾಡ್ಬೇಕಾದ್ರೆ.
ಓಕೆ, ದೇರ್ ಇಸ್ ಓನ್ಲಿ ಒನ್ ಮೈಕ್ ದೆನ್.
ಎರಡು ಮೈಕ್ ಅದಾವ್ರಿ, ಇದು ಒಂದು ಮೈಕ್ ಪ್ರಾಬ್ಲಮ್ ಆಯ್ತು.
ಇವತ್ತಿಗೆ ಬಾಯ್ಸ್ಗೆ ಕೊಡಿ. ಏನಿಲ್ಲ, ಐ ಆಮ್ ನಾಟ್ ಗೋಯಿಂಗ್ ಟು ಟ್ರಬಲ್ ಯು. ಡೋಂಟ್ ವರಿ ಬಾಯ್ಸ್, ಡೋಂಟ್ ಬಿ ಸ್ಕೇರ್ಡ್. ಐ ಜಸ್ಟ್ ವಾಂಟ್ ಯು ನೋ ಹೌ ವೆಲ್ ಯು ಆರ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡಿಂಗ್ ದ ಕಾನ್ಸೆಪ್ಟ್ ಅಷ್ಟೇ. ಓಕೆ, ಚಿಕ್ಕೋಡಿ ಕ್ಲಾಸ್ 21. ಕ್ಲಾಸ್ 21 ಪ್ಲೀಸ್. Where is your mic? Where is your mic? Speak. Archana ma'am, ask them to speak.
Yes ma'am.
Girls, ನಿಮ್ಮ ಹತ್ರ ಒಂದು ಮೈಕ್ ಇದೆ ಅಲ್ಲ, ಇನ್ನೊಂದು
Yes,
ಮೈಕ್
ma'am.
ಇದೆ. Where is one more mic? Only one mic?
No ma'am.
Boys, take the mic. ಸಾಕು. Now, no girls will speak, okay? Boys, are you able to understand what we are doing? I'm talking to only boys. Chikkodi boys, Dharwad boy. Are you understanding what we are doing?
Yes, ma'am.
Okay, fine. Then give me the answer. What does
Yes,
this
ma'am.
I gene ಏನು ಮಾಡುತ್ತೆ? What is I gene coding for? Sorry.
Protein.
Every gene codes for protein, dear. Which protein? ಅಲ್ರೋ ಪಾಸರೇ ಆಗ್ತಿರಲ್ಲ, ಡೆಫಿನೆಟ್ಲಿ ಪಾಸ್ ಆಗ್ತಿರಲ್ಲ.
Repressor protein. Yes, ma'am.
Yes, it codes for repressor. Fine. What is this O gene? This O.
Operator.
Are you a boy? Anyone from boys? What is O gene? Hurry up, don't waste my time.
Operator.
Okay. What is this Z gene?
Synthesis of beta galactosidase.
Yes.
Beta
Who is
galact
this?
beta.
Who is this? Good one. ಯಾರು ಹೇಳೋದು? Which batch? ಚಿಕ್ಕೋಡಿ which batch? ಹೆಸರು ಹೇಳ್ರು.
Beta galactosidase protein.
ಹಾ ಗೊತ್ತಾಯ್ತು. ನೀನು ಯಾವ ಕ್ಲಾಸಿಂದ ಮಾತಾಡ್ತಾ ಇದೀಯಾ? Which class?
ಚಿಕ್ಕೋಡಿ 20 ಇಂದ ಮಾತಾಡೋದು.
ಹೇಳೋಕೆ ಏನು ತ್ರಾಸ ಸರ್ ಇವರಿಗೆ? ವಾಟ್ಸ್ ಪ್ರಾಬ್ಲಮ್ ಟು ಟೆಲ್ ಮಿ ದ್ಯಾಟ್ ಆಲ್ಸೋ? ಇಟ್ ಈಸ್ ರೈಟ್ ಆನ್ಸರ್. ಐ ಆಮ್ ಅಪ್ರಿಷಿಯೇಟಿಂಗ್ ಯು. ಹೊಗಳಿದ್ರು ಪ್ರಾಬ್ಲಮ್, ಬೈಸ್ಕೊಂಡ್ರು ಪ್ರಾಬ್ಲಮ್. ಏನು ಮಾಡ್ಬೇಕು ನಿಮ್ಮ ಜೊತೆ? ನಿಮ್ಮ ಜೊತೆ ನಾನು ಇಲ್ಲ, ಓಕೆ? ಇಲ್ಲಾಂದರೆ ಅಲ್ಲಿದ್ದರೆ ಹೇಳ್ತಿದ್ದೆ. ಗ್ಯಾಲಕ್ಟೋಸಿಡೇಸ್. ದಿಸ್ ಈಸ್ ಬೀಟಾ ಗ್ಯಾಲಕ್ಟೋಸಿಡೇಸ್. ಐ ವಿಶ್ ಐ ವುಡ್ ಮೀಟ್ ಯು ಒನ್ಸ್, ಓಕೆ? ಪ್ಲೀಸ್ ಪ್ರೇ ಫಾರ್ ದ್ಯಾಟ್. ಪೂಜಾ ಮ್ಯಾಮಿಗೆ ಭೇಟಿ ಆಗ್ಬೇಕಂತ. ವೈ? ಕುತುಬ್ ಮಿನಾರಿಗೆ ಬರ್ತಿರಲ್ಲ ಅವಾಗ ನಂಗೆ ಭೇಟಿಯಾಗಿ. ವೈ ಜೀನ್ ಬಾಯ್ಸ್ ಕ್ಲಾಸ್ 21. ವೈ ಜೀನ್ ಕೋಡ್ಸ್ ಫಾರ್ ಹರಿ ಅಪ್ ರೇ.
Permease.
ಓಕೆ. ಆ್ಯಂಡ್ ಉಳಿದಿದ್ದು
Resistance.
ಟ್ರಾನ್ಸ್ ಅಸಿಟೈಲೇಸ್. ಯು ಆಲ್ ಆರ್ PU2 ಬ್ಯಾಚ್ ಅಲ್ಲ? PU1 ಅಲ್ಲಿ ನರ್ವಸ್ ಸಿಸ್ಟಮ್ ಯು ಹ್ಯಾವ್ ಡನ್ ನರ್ವ್ ಕಂಟ್ರೋಲ್ ಅಂಡ್ ಕೋಆರ್ಡಿನೇಷನ್. ರಿಮೆಂಬರ್?
Yes, ma'am. Yes, ma'am.
Now when I'm writing by left hand, which part of my brain is becoming more active?
Right.
Sure?
Yes, ma'am.
ಅದೇನ್ ಕ್ರಾಸ್ ಆಗತ್ತೋ? ರೈಟ್ ಸೈಡ್ ಶುಡ್ ಬಿ ರೈಟ್ ಸೈಡ್, ಲೆಫ್ಟ್ ಸೈಡ್ ಶುಡ್ ಬಿ ಲೆಫ್ಟ್ ಸೈಡ್. ಲೆಫ್ಟ್ ಸೈಡ್ ರೈಟು, ರೈಟ್ ಸೈಡ್ ಲೆಫ್ಟು. ಕ್ರಾಸ್ ಕನೆಕ್ಷನ್ ಹೆಂಗೆ? Okay, that is correct. Be confident on your answer. Fine. Now we have wrote what are the genes and what are their respective products. Now imagine very first that the whole bacteria, now pay attention, whole bacteria relies on the source of energy. Energy source elinda baratte? You eat something and from that you get energy. So for bacteria also has to eat something. Something what? Glucose, galactose, lactose, fructose. These are the different carbohydrate source from which a bacteria can take energy. Chikodi class 21 boys. ನನಗೆ ಬೇಕಾಗಿಲ್ಲ ಇದು, ನನಗೆ ಹರ್ಕತ್ತಿಲ್ಲ. ಲೆಟ್ ಮಿ ಟೆಲ್ ಯು ವೆರಿ ಕ್ಲಿಯರ್ಲಿ, ಡೋಂಟ್ ಮೇಕ್ ಮೀ ಶೂಟ್ ಮೈ BP up, okay? I'll literally stop the whole class. This is not my need. So now when this energy source is taken inside the cell, if you remember glycolysis ಮಾಡಿದ್ವಿ, respiration in PU1. ನಾನು ಮಾಡಿಸಿಲ್ಲ, but you have done na respiration chapter, you have done glycolysis, you have done, where glucose ತಗೊಂಡು ATP ಮಾಡುತ್ತೆ cell. Every cell will do that. So which means the energy source is required. Generally bacteria will also use glucose only as an energy source, but whenever glucose is not available in the medium or in the environment, suppose let's say glucose is not available, at that time what will happen? This lactose or arabinose or different alternative source of energy is being used. ಪ್ರತಿಯೊಂದು ಎನರ್ಜಿ ಸೋರ್ಸಿಗೆ ಬೇರೆ ಬೇರೆ ಜೀನ್ಸ್ ಇದೆ, ಬೇರೆ ಬೇರೆ ಒಪೆರಾನ್ ಮಾಡೆಲ್ ಇದೆ. So in your book they have given only about lactose operon. So let us see what happens in the presence and in the absence of lactose. So if I talk the first case, when the lactose is absent or not there, when lactose is not there, at that time what will happen? This repressor or the regulator gene is going to make what protein? Yes,
mRNA
girls, you can speak now.
mRNA.
mRNA and final product is what? Repressor protein.
Repressor protein.
this protein will go and sit on the operator region. Correct? Idrinda ಏನಾಯ್ತು? What happened? Because of this, the DNA, the RNA polymerase cannot synthesize. RNA polymerase recognize the promoter region. What is the function of promoter? RNA polymerases recognize, even in transcription we saw na, there is a promoter, then there is a gene, and there is a terminator. Promoter yak beku, it's one kind of a signal. It's a recognition sequence for RNA polymerase. Promoter region, the RNA polymerase will recognize it and start synthesizing the respective genes. But now the problem is, even if the polymerase recognizes the promoter and synthesizes, because of this operator present in between, it cannot move forward. And that is why all these three things will not be synthesized. Illivargu clear? Any doubts?
No, ma'am.
Now, in second case, suppose there is lactose present. So what will happen? Lactose bantantandre, lactose will bind where? Where will the lactose bind? Suppose this was your repressor. Now in the medium lactose has come. This lactose will come and bound on this repressor protein, which brings about the conformational changes in this repressor protein and falls off. Repressor protein will fall off, due to which now this polymerase can transcribe the all three genes. And then we say that this operon is on, and here this operon is off. So in presence of lactose, because the operon is on, we say it is a inducible operon. I'll write it here only. Lac operon is a inducible operon, or lactose here acts as inducer. Clear, Illivargo?
Yes, ma'am. Yes, ma'am.
Now, why you need Z gene, Y gene, and A gene? This Z gene is going to make the beta galactosidase, which is required for what? Beta galactosidase ಏನು ಮಾಡುತ್ತೆ? This will
On and off.
ಹಂಗಲ್ಲ, hydrolyzes or cleaves lactose into
Glucose and galactose.
glucose and galactose. And then finally glucose and galactose can be used for the energy purpose. Glycolysis ಮಾಡ್ಬಿಟ್ಟು the cell can make ATP out of it. So lactose is called as disaccharide. Now those who wants to aim for NEET, right, remember this, that lactose is a disaccharide. Disaccharide meaning having two monomers. Two monomers ಸೇರಿ one molecule ಆಗಿದೆ. ಈ ಥರ if there are three monomers, we say trisaccharide, tetrasaccharide, or if there are more than 3 to 4, then we say a polysaccharide, a big molecule. For example, DNA is made up of single monomer of nucleotide. ಒಂದೊಂದೊಂದೊಂದು ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಸೇರಿ ಒಂದು ದೊಡ್ಡ DNA ಆಗಿರುತ್ತೆ. ಹಾಗೆ ಒಂದೊಂದೊಂದೊಂದು monomer ಸೇರಿ ಒಂದು ದೊಡ್ಡ polymer ಆಗುತ್ತೆ. Like that, this is not a polymer. Polymer andre more than 2 to 3, 8 to 10 nucleotides ಜಾಸ್ತಿ ಆತಂದ್ರೆ we say a polymer, big molecule. When it's only made up of 2, then we say it's a disaccharide. So lactose directly consume madak agalla. Lactose cannot be consumed directly. You cannot, or any cell cannot. Any cell has to degrade or has to cleave the, suppose you eat chapati or rice, right? It's a carbohydrate. Chapati ಹೆಂಗಿದ್ದಂಗೆ ಒಳಗೆ ಹೋಗುತ್ತಾ? No, that would be chopped, and first of all you will do digestion, correct? Digestion ಆದಮೇಲೆ in stomach, in intestine, whole chapati carbohydrate from that, gluten from that whole chapati would be broken down finally to give you a glucose. And that glucose, with the help of blood, goes to the different cells of your body, which means the big complex carbohydrate has to be cleaved down so that cell can take that and make a energy out of it. This is applicable to bacteria also. It cannot take larger carbohydrates, so ಒಳಗೆ ತಗೊಂಡ್ಮೇಲೆ it has to cleave it. Cleave ಮಾಡ್ಬೇಕಂದ್ರೆ you have to have that enzyme. So beta galactosidase helps to cleave this. Now permease, this permease helps to permit or allow the lactose to come inside the cell. Why? Because all this energy is present outside the medium. A cell ಹೊರಗಡೆ ಇರುತ್ತೆ. We are talking about prokaryotes, right? Bacteria are found everywhere in the environment. Environment ಸುತ್ತಮುತ್ತಲ ಏನು nutrient ಇದೆ, that nutrient bacteria will keep on taking. We eat food because we are higher eukaryotes. We can eat food by our hand. For bacteria, what is food? Anything which is available in the medium or environment, that itself becomes a food for bacteria. So that has to go inside the cell. So when lactose has to go inside the cell, it needs the help of enzyme permease. This is clear? Illivargu clear? Any doubts, please ask. Don't hesitate. Nan baitini ant heli, don't hesitate. Baiskori, but still your concept will get clear. And lastly, A is trans-acetylase. This is actually involved in detoxification. Most the things which are unwanted would be removed by this acetylase. This is clear. On and off regulation, what happens in the presence of lactose. Go through the book. On certain nodkoli, I'll give you 2 minutes time. Go through the book, check what is not understood, check if I have missed something or anything, and ask. The Harvard girls, is it clear? At least yes or no. It's a no, then no problem. We can repeat it. Pranamendu girls, what about you?
Yes, ma'am.
Because questions ಮಾಡಿದಾಗ you will make mistakes then. No? Not understood? Anything? Chikodi class 20, class 22 girls? Okay, nodkoli. Check once. If anything is not understood, ask. Good to go? ಮಾತಾಡ್ರು ಇಷ್ಟು ಸೈಲೆಂಟ್ ಆದ್ರೆ
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
ಶ್ಯೂರ್ಲಿ
Yes, ma'am. Yes, ma'am.
Yes, ma'am.
ಓಕೆ
Yes, ma'am.
ನಾನೇ ಕ್ವಶನ್ ಕೇಳ ವಿಲ್ ಇಟ್ ಬಿ ಓಕೆ ವಿಲ್ ಯು ಬಿ ಏಬಲ್ ಟು ಆನ್ಸರ್
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
okay
Yes,
why
ma'am.
lack operon is called as inducible operon? One of you raise your hand. Inducible operon ಅಂತ ಯಾಕೆ ಹೇಳ್ತೀವಿ?
Because it depends on presence of lactose, which acts as inducer.
It ಅಂದ್ರೆ ಏನು? What depends? That is important. What depends?
Lactose. Lactose.
ಇನ್ನೊಮ್ಮೆ ಹೇಳು. What I asked, why it is called inducible operon? It depends upon the presence and absence of lactose, you told, right? It means what? It means what? What
Operon.
depends? What depends upon the presence of lactose and
Operon.
Operon.
okay. ಇದನ್ನ ಸಿಂಪ್ಲಿಫೈ ಮಾಡಿ ಹೇಳ್ತೀನಿ. Instead of saying operon, the synthesis or the regulation of these genes depends upon the presence and absence. Will it be more appropriate? ನೀವು ಹೇಳೋದು ಕರೆಕ್ಟ್ ಇದೆ, but put it into this words. You can get more marks like this. Okay? This is a small tricks. Okay, fine. Then, who helps the lactose to come inside the cell? Who helps the lactose?
Repressor.
Why?
Permease.
Repressor.
Repressor.
Permease.
Okay, repressor ಯಾರು ಕೊಟ್ಟಿದ್ದು? Which batch
Madam.
answered me repressor? Come
Madam.
on, say. Who answered me repressor? Radamenur, is it you?
Yes, ma'am.
Yes,
Okay,
ma'am.
repressor protein ಏನು ಮಾಡುತ್ತೆ? No problem. What does repressor protein do?
RNA polymers.
Binds to operator.
RNA polymers.
Permease. You were not audible, ma'am. Ma'am, you are not audible. Not audible. Ma'am, you are not audible.
Not audible. Sound ಕೇಳಿಸಿ ಬರತ್ರಿ ಮ್ಯಾಮ್.
You are not audible. Not audible.
No, ma'am.
Not audible, ma'am.
Not audible. Not audible. Yes, ma'am.
Yes, ma'am.
Yes,
No,
Yes,
ma'am.
ma'am.
Yes, ma'am.
Yes,
No,
ma'am.
ma'am.
Yes.
Hello, can you hear?
Yes,
ma'am.
ma'am.
Yes,
ma'am.
Yes, ma'am. Yes, ma'am.
Okay, fine. Maybe some technical error. Okay. Fine. Okay, on that question ಮಾಡೋಣ. Let us see if you can answer. Are you sure na this
Ma'am.
concept is clear?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Okay. Then tell me one more. I think one thing left out. What is this?
Yes, ma'am.
What is this? What is negative regulation of operon?
Lack operon by lack operon by the
Lack operon by
refer
repressor.
to the negative regulation.
Read it again. Lack operon.
Regulation of lack operon by
Repress refers
repressor.
to the negative regulation.
How? How? The lack operon is an example of negative regulation. What do you mean by that?
Under control the positive.
ಹೆಂಗೆ ಇದು ನೋಡು ಹೆಂಗೆ ನೆನಪಿಟ್ಕೋಬೇಕು ಗೊತ್ತಾ ನೆಗೆಟಿವ್ ರೆಗ್ಯುಲೇಷನ್ ಮೀನ್ಸ್ ವಾಟ್ ಈಗ ಇಫ್ ಐ ವಾಂಟ್ ಟು ಡಿಕ್ರೀಸ್ ದ ಸ್ಪೀಡ್ ಆಫ್ ಅ ಫ್ಯಾನ್ ಏನು ಮಾಡ್ತೀನಿ ನಾನು ಐ ವಿಲ್ ರೋಟೇಟ್ ಇಟ್ ಇನ್ ದ ಆಂಟಿ ಕ್ಲಾಕ್ ವೈಸ್ ಹಿಂಗೆ
Yes, ma'am.
ರೋಟೇಟ್ ಮಾಡ್ತೀನಿ ಇಫ್ ಯು ವಾಂಟ್ ಟು ಇನ್ಕ್ರೀಸ್ ಪಾಸಿಟಿವ್ ದೆನ್ ಯು ವಿಲ್ ಡೂ ಇಟ್ ಅಸ್ ಅ ಕ್ಲಾಕ್ ವೈಸ್ similar concept if I want now negative regulation repressor acts as a negative regulator here repressor acts as a negative regulator hangantandre ಏನು whenever repressor protein is present this protein will stop the synthesis of all these three genes that is called negative regulation.
Regulation.
When repressor is not there it will on the all three genes. Now same concept if I ask what is lactose doing, lactose is doing negative regulation or positive regulation. Repressor ಬದಲು ಇಲ್ಲೇ lactose ಅಂತ ಇಟ್ಕೊಳ್ಳಿ. So lactose will do negative regulation or positive regulation?
Positive.
Positive
Correct.
regulation.
Positive, which means the presence of lactose will switch on the gene, absence of lactose will shut down the gene. Okay. Fine. ನೋಡೋಣ. Let me see if you can do few questions. Cue cards, please.
Option A, ma'am.
Okay. Scan the
Ma'am,
answers.
cue
Poll
call
has
is
been
negative.
started.
Option A, ma'am.
Okay. A and D, both options. What they're asking, regulation of gene expression be exerted at which processes the gene can be regulated. Regulated means it can be on or it can be off. Transcription level also, yes. Post transcription level, yes. Translation level, yes. All of this, at all of the three processes you can switch on and switch off the gene. ಮೂರು ಟೈಮಿಗೆ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್, post transcription and translation, at all the three steps the process of gene regulation can be done. Suppose ಇಲ್ಲಿ ಮಾಡೋಕ್ಕಾಗಲಿಲ್ಲ ಅಂದರೆ ಅಟ್ಲೀಸ್ಟ್ ಅಟ್ ಪೋಸ್ಟ್ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಲೆವೆಲ್ ಯು ಕ್ಯಾನ್ ಸ್ಟಾಪ್ ದ ಜೀನ್. ಸಪೋಸ್ ಇದನ್ನು ಮಿಸ್ ಆಯಿತು ಅಂದರೆ ಟ್ರಾನ್ಸ್ಲೇಷನ್ ಲೆವೆಲಲ್ಲಿ ಯು ಕ್ಯಾನ್ ಸ್ಟಾಪ್ ದ ಜೀನ್. ಸೊ ದೀಸ್ ಆರ್ ದ ತ್ರೀ steps at which transcription can be stopped or regulation of the whole gene can be controlled. All the three is correct. Majority of the people has given me option A as the answer. No, all three are correct. I hope you got your right answer. Shall I take the names now who has given me the wrong answer? ನಿಮಗೆ ಗೊತ್ತು ನೀವೇನು ಆನ್ಸರ್ ಮಾಡಿದ್ದೀರಾ ಅಂತ. Who gave this what is this lactose operant? Direct answer kodi, don't scan the cards.
Polycystonic.
D, ma'am.
Option
Option C.
C. Why option C? ಸುಮ್ಮನೆ ಹೇಳಬೇಕಂತ ಹೇಳಬೇಡಿ. Polycystonic, it is given in the definition itself. ನಾನು ಇಷ್ಟೋ ತನಕ ಹೇಳಿದೆ, it is a polycystonic. Any confusion here? You want me to teach it again?
No, ma'am.
Who gave the operon model?
Option C, ma'am.
Option C.
Option C.
Jacob and Monad.
Option.
ಇದನ್ನ ಮಾಡಿ. Scan now. Please scan it. No answers, okay? Don't give answers. Let me see. Start the poll. Start the poll. Where is he? Click here. Click here. ಇಲ್ಲಿ ಕ್ಲಿಕ್ ಮಾಡಿ.
Option B.
What?
Option B.
Permease.
B. Why? Why permease?
Option A, ma'am.
These are previous.
Option A, ma'am.
Okay. Can anyone explain me this? Okay. Option A ಯಾರಾದರೂ ಹೇಳ್ಬೋದಾ? Can anyone explain me this? Can anyone explain me option A answer ಯಾಕೆ correct ಅಂತ? Come on. No?
Goes into galactocytes and glucose.
Because it cleaves the lactose into galactose and glucose. Okay. Option B ಯಾರು ಆನ್ಸರ್ ಕೊಟ್ಟಿದ್ದೀರಾ? Can you tell me why B?
Permease allows the entry of lactose in bacteria.
Who is this?
Chikkodi.
ಯಾವ class? Tell me your name.
Class 20. Riya.
Priya. Okay.
Riya.
So in a growth medium, Priya only, right? No?
Riya.
Riya. Okay. Fine. Sorry. Lactose is added in the medium. The lactose is transported inside the bacteria by. Why galactosidase? Galactosidase is required once the lactose is coming inside the bacteria. To come inside the bacteria, the permease is required. Idin ಹೇಳಿದೆ ನಾನು. Permease is required that will help the lactose to come inside the cell, and once it is coming inside the cell, then the beta galactosidase will cleave this lactose into glucose and galactose. Is it clear? So permease is the right answer. Anybody, any doubt?
No, ma'am.
ಇವತ್ತು ಗೊತ್ತಾಗಲ್ಲ. ಐ ಆಮ್ ಗೋಯಿಂಗ್ ಟು ಸೆಟ್ ಅ ಪೇಪರ್ ಆನ್ ದಿಸ್ ಅಲ್ಲ, ಆವಾಗ ನೋಡ್ತೀನಿ ಯಾರ್ಯಾರು ಹೆಂಗೆ ಮಾಡ್ತೀರಾ ಅಂತ. ಹಾಂ, ಅಗೇನ್ ರೇಸ್ ದ ಕಾರ್ಡ್ಸ್. Come on, raise the cards, people.
Option B.
Wait, let me get the responses. Wait.
Option B, ma'am.
Okay. Everybody's answering A, B, C, D. ಏನು ಹೇಳ್ತಾ ಇದೆ? In lac operon, if mutation occurs in the middle gene of the structural gene. Middle gene ಯಾವುದು?
Y.
Y. Okay, so what will happen? Beta galactosidase will not, connection ಇಲ್ಲ. Direct answer.
Yes, B, ma'am.
Then why there are so many people answering me A and C? Select C option. There are so many people giving me C option also. Why? Not understood? Not understood, people? ಇದನ್ನು ಮಾಡಿ. Think, okay? This is an important question. It's a tricky question. Think. Think and give me the answers. You can tell me directly. ಹಂಗೆ ಡಿಸ್ಕಸ್ ಮಾಡ್ಬೋದು ನನ್ನ ಹತ್ರ. You can talk to me directly.
Option C, ma'am.
Okay. Why? Why you think so? Option C? Come on, discuss. It's an open discussion. You need to discuss to me. Why you think option C? ಮತ್ತೆ ಯಾರಾದರೂ ಇದರ ಆನ್ಸರ್ ಹೇಳ್ತೀರಾ? In operon model, the regulator gene regulates the chemical reaction in the cell by a regulator gene. How it is going to regulate the whole synthesis? Inhibiting the transcription of mRNA, inhibiting the enzymes in the reaction, inhibiting the substrate in the reaction, inhibiting the migration of mRNA into cytoplasm. Repressor gene, how it is going to regulate the lac operon? Talk. If you don't understand, say no. But sitting silent, I'll get struck. Don't sit silent. Random in class 1. Anyone can answer? Mic ಎಲ್ಲಿದೆ? Huh. Can you answer this or discuss with this? Discuss to me. Anyone? Not a one single girl can answer me. ಯಾರಿಗೂ ಗೊತ್ತಿಲ್ಲ. Class 2 ರಾಣೆಬೆನ್ನೂರ್. ನೀವು ಆಪ್ಷನ್ ಹೇಳಿದರೆ ಅದನ್ನು ಯಾಕಂತ ಹೇಳಬೇಕು. ಸಿ ವೆನ್ ಯು ಆನ್ಸರ್ನ ಐ ವಿಲ್ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ವೆದರ್ ಯು ಗಾಟ್ ದ ಕಾನ್ಸೆಪ್ಟ್ ಕರೆಕ್ಟ್ ಆರ್ ನಾಟ್. ದಿಸ್ ವಿಲ್ ಹೆಲ್ಪ್ ಮಿ ಟು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್. ಸೊ ಯು ಹ್ಯಾವ್ ಟು ಹೆಲ್ಪ್ ಮೀ. ಧಾರವಾಡ, ಟ್ರೈ.
Ma'am,
Propanol consists of
The
the one regulator
pressure protein,
gene.
Wait. Two people are speaking. ಎಲ್ಲಿಂದ ಮಾತಾಡ್ತಿದ್ದೀರಾ? Who is
from
speaking?
Dharwad.
ಹಾ, ಧಾರವಾಡ. Okay, tell me.
Ma'am, in the absence of the inducer, the repressor protein will binds to the operator region and inhibits the process, ma'am. Hence, the Z gene will not able to synthesis beta galactosidase enzyme. Hence, lactose will not be produced. Hence, option C is right.
Lactose will not be produced ಅಲ್ಲ.
Means beta galactosidase enzyme is not produced, ma'am.
ಹಾ.
Hence, the beta galactosidase is not able to break down the lactose into glucose and the galactose.
Okay, ಹಾ.
So, option C is right.
ಬಟ್ ಇಲ್ಲಿ ಏನು ಹೇಳ್ತಾ ಇದೆ ಆಪ್ಷನ್ ಸಿ, ಇನ್ಹಿಬಿಟಿಂಗ್ ದ ಸಬ್ಸ್ಟ್ರೇಟ್ ಇನ್ ದ ರಿಯಾಕ್ಷನ್. ಇಲ್ಲಿ ಸಬ್ಸ್ಟ್ರೇಟ್ ಯಾರಿಗೆ ಕನ್ಸಿಡರ್ ಮಾಡ್ತಾ ಇದ್ದೀರಾ?
Lactose.
Lactose. Acha. Okay, fine. This is the one way. What's your name, dear?
Renuka, ma'am.
Renuka. Okay. Good logic. Fine. Anyone else has any other answer? Anyone else has any other answer? Chikodi class 2021. ಅಲ್ಲ, ಕ್ವಶನ್ ಅರ್ಥ ಆಗಿಲ್ಲ ಅಂತಂದ್ರೆ ಹಂಗೆ ಹೇಳಿ, ಮ್ಯಾಮ್ ವಿ ಹ್ಯಾವ್ ನಾಟ್ ಅಂಡರ್ಸ್ಟುಡ್ ಕ್ವಶನ್ ಇಟ್ ಸೆಲ್ಫ್, ಐ ವಿಲ್ ಟೆಲ್ ಯು ವಾಟ್ ಇಟ್ ಮೀನ್ಸ್.
Sir,
ಏನಾದರೂ ಹೇಳ್ಬೇಕು ತಾನೆ?
This is the one regulator gene.
See, what regulator gene is going to regulate the chemical reaction? Regulator gene ಏನು ಕೊಡುತ್ತೆ? It synthesizes
Repressor protein.
correct. So repressor protein will go and sit on the operator region, and therefore this blocks the, simply transcription will get blocked. Because transcription is blocked, there is no further synthesis of any enzyme. So inhibiting the transcription of mRNA is more appropriate. Inhibiting the enzyme, repressor ಹೋಗಿ ಯಾವುದು ಎಂಜೈಮಿಗೆ ಅಟ್ಯಾಚ್ ಆಗಲ್ಲ. Inhibiting the substrate, Renuka, you said lactose is a substrate, correct, but repressor is not joining with the substrate alla. That's why we cannot consider this. Your logic or your answer was half correct. But lactose and repressor, do they combine? No, lactose will not combine. Repressor will go and combine on the operator region. ಇಲ್ಲೇ ರೆಗ್ಯುಲೇಷನ್ ಮಾಡೋದ್ರೆ ನಮಗೆ ಆಫ್ ಮಾಡೋದ್ರೆ, ವಿ ಹ್ಯಾವ್ ಟು ಆಫ್ ದ ಜೀನ್. ಲ್ಯಾಕ್ಟೋಸ್ ವಿಲ್ ಆನ್ ದ ಜೀನ್. ಸೊ ನೀನು ಏನು ಲಾಜಿಕ್ ಹೇಳಿದೆ ಆ ಪ್ರಕಾರ ಲ್ಯಾಕ್ಟೋಸ್ ಒಳಗೆ ಬರಬೇಕಂದ್ರೆ ಆ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಆಗಬೇಕು. ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಆಗಲಿಲ್ಲ ಅಂದರೆ ಪರ್ಮಿಯೇಸ್ ಇರಲ್ಲ. ಪರ್ಮಿಯೇಸ್ ಇಲ್ಲ ಅಂತಂದರೆ ಲ್ಯಾಕ್ಟೋಸ್ ಒಳಗೆ ಬರಲ್ಲ. ಮತ್ತು ಲ್ಯಾಕ್ಟೋಸ್ ಒಳಗಿಲ್ಲ ಅಂತಂದರೆ ಹೌ ದಿಸ್ ರಿಪ್ರೆಸರ್ ವಿಲ್ ಬೈಂಡ್ ಟು ಸಬ್ಸ್ಟ್ರೇಟ್? ಆಗಲ್ಲ.
Yes, ma'am.
Correct. So this also not possible. Then inhibiting the migration of mRNA, no question of migration. Operon is operating at prokaryotic level. Cytoplasm and nucleus each are separate alla. Prokaryotes alli there is no well defined nucleus. Everything is happening in the whole big one cell. So migration is out of question. I mean out of the logic. Understood? So repressor gene actually inhibits the transcription of mRNA. That is what we did. It is a twisted question actually. But yell hotro. Repressor gene is actually inhibiting the whole transcription. Got it?
Yes, ma'am.
See people, ಕೆಲವೊಂದ್ಸತೆ ಬರೀ ಥಿಯರಿ ಮಾಡೋದು ಇಂಪಾರ್ಟೆಂಟ್ ಅಲ್ಲ. ವೆನ್ ಯು ಆರ್ ಗಿವಿಂಗ್ ಕಾಂಪಿಟೇಟಿವ್ ಎಕ್ಸಾಮ್, ಸಾಲ್ವಿಂಗ್ ಕ್ವಶನ್ಸ್ ಇಸ್ ಮೋರ್ ಇಂಪಾರ್ಟೆಂಟ್ ದೆನ್ ಡೂಯಿಂಗ್ ಥಿಯರಿ. ಅಫ್ಕೋರ್ಸ್ ಥಿಯರಿ ಗೊತ್ತಾದ್ಮೇಲೆ ತಾನೇ ಯು ಕ್ಯಾನ್ ಡೂ ದ ಕ್ವಶನ್ಸ್, ಐ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ದ್ಯಾಟ್. ಬಟ್ ಮೆನಿ ಅ ಟೈಮ್ಸ್ ವೆನ್ ಯು ಡೂ ಕ್ವಶನ್ಸ್, ನೋ ಕಾನ್ಸೆಪ್ಟ್ಸ್ ಗೆಟ್ ಆಟೋಮೆಟಿಕ್ಲಿ ಸಾಲ್ವ್ಡ್. This helps. So trust me, try to do more and more questions. Your concepts will automatically get cleared. Your concepts will get automatically be perfect in that. Fine. Okay, there are few more questions. You know, HGP and DNA fingerprinting. I wanted to finish whole concept today only, but fine, we could not do it. Tomorrow's class, you will read and come. HGP and fingerprinting is only theory. Points de bare. Adralli concept. Chikode people, did I finish the class? Is this the way to behave? You're closing your books and packing your bags? Salpa adru sense ilva nimage? I'm standing here right in front of you with my hand broken. I'm doing this class for you people, not for me. tomorrow I want all of you to read and come HGP and DNA fingerprinting. It is not much concept based. ಕೆಲವೊಂದು ಕಾನ್ಸೆಪ್ಟ್ ಅಷ್ಟೇ ಇದೆ, ವಿ ಎನ್ ಟಿ ಆರ್ ಆ್ಯಂಡ್ ರಿಪಿಟೇಟಿವ್ ಸೀಕ್ವೆನ್ಸಸ್ ಆನ್ ಅಷ್ಟೆಲ್ಲ ಹೇಳ್ತೀನಿ, ಬಟ್ ವಿ ವಿಲ್ ಸಾಲ್ವ್ ಮೆಜಾರಿಟಿ ಕ್ವಶನ್ಸ್ ಆನ್ ದ್ಯಾಟ್. ಕ್ಲಿಯರ್? ರೀಡ್ ಆ್ಯಂಡ್ ಕಮ್, ಡೋಂಟ್ ಬಿ ಲೈಕ್ ಅ ಡಮ್ ಪೀಪಲ್ ನಾವು. ಯು ಆರ್ ಇನ್ PU2. ಬೆಳಗಾದ್ರೆ ಎಕ್ಸಾಮ್ ಬಂದಿದೆ. ಥ್ಯಾಂಕ್ ಯು. End the class. I don't want to.
Bye, ma'am.
Thank you, ma'am. Thank you, ma'am.